I'd like to introduce at this stage Dominique Anderson. She is a senior researcher at the South African National Bioinformatics Institute. And Dominique is going to talk to us about how the functionality within Baobab LIMS has evolved in the context of metadata standards. Thank you, Dominique.
Hi, everyone. I hope everyone can see my screen.
Yes, we can.
Okay, great. So thank you very much for that introduction, Alan. I really appreciate it. And thank you very much to the organizers for the opportunity to present this talk. So what is metadata? Quite simply put, metadata is data about data. It is the associated information that is required to understand and effectively use data. And it is a key to unlocking a resource. Metadata standards therefore establish a consistent way of structuring and understanding data to ensure proper use and interpretation. A metadata standard increases the utility of the data. It supports interoperability, makes it easier to find the data, provides access to the data, and enhances discovery from the data through its reuse. Metadata management, therefore, is the administration of data. It determines processes of how data is integrated, accessed, shared, linked, analyzed, and maintained. The management applies to physical and virtual data formats, and resources are consumed in this task to maximize the potential of an organization's data. In 2006, Hamby coined the term, "Data is the new oil." Michael Palmer then expanded on Hamby's quote, saying that, like oil, data is valuable, but if unrefined, it can't really be used. So metadata management turns raw data into information. And clearly, inconsistencies in data management leads to inconsistencies downstream and compromises the integrity and the value of that data. So I'm going to talk about laboratory operations specifically. So data management is improved by using a tool such as the Laboratory Information Management System. This increases laboratory potential. So if we think of a sample entering the laboratory, that sample is a form of data. The associated metadata, so data of that data, enters the LIMS. The LIMS effectively draws the roadmap for the sample lifecycle, and this creates a valuable linkage between the sample and its associated data. Therefore, standardizing the data makes the roadmap readable in a universal way, and harmonization is incredibly beneficial to interoperability. The Baobab LIMS is an open source LIMS. It was developed using the SOPs of a functional biocollection facility in Cape Town. This biocollection facility is focused on the collection of human biospecimens. And as such, the first metadata standard which was incorporated into the system was the minimum information about biobank data sharing. This included standard minimum metadata related to sample donors, disease ontology, anatomical site terms and descriptors, which could all be linked to that sample. This data would specifically facilitate finding of an appropriate resource and subsequent sharing of that resource. In 2020, SARS-CoV-2 literally went viral, and this public health emergency has resulted in a global combined effort. Baobab LIMS had to evolve, and we saw the need to incorporate standardization into the system for infectious disease samples. The PHASE Consortium established the SARS-CoV-2 contextual data standard, and Baobab LIMS opted to incorporate this standard. We did this so that appropriately structured and consistent metadata could be captured in relation to SARS-CoV-2 research. This evolution of Baobab LIMS aims to promote material and data reuse and sharing through interoperability. The loss of information content, also known as information entropy, generally increases over archival time. And by incorporating these metadata fields in the LIMS, we hope that we increase sample and data longevity. Before we could incorporate the data specifications, we had a number of things to consider, and some items were already on our radar from previous experience, as well as valuable retrospective user input. Fields that were mandatory had to be added, but we had to evaluate the other fields to ensure the suitability for a sample LIMS. Baobab LIMS is not an NGS LIMS, and as such does not contain the quality metrics associated with sequencing and bioinformatics. However, we had to ensure some overlap because these downstream processes are in fact linked to the sample management processes. We needed to keep the existing architecture of Baobab in mind. Baobab has process flow and default fields essential to function, which still need to be in place. We needed to remember that sample numbers would be high and that infrastructure capacity is not the same in every single setting. We needed simplicity without compromising the goal of standardization. Once we drafted our plan, we began coding, testing, and in some cases, we had to make modifications. Another consideration that we had to take was making sure that this was applicable in a post-COVID world. So in essence, making it more sustainable and future-proof. The fields incorporated into Baobab LIMS consist of configurable content as well as hard-coded options. The configurable content relates to fields which could be applicable to other viral diseases. And once this data is configured in the system, a simple drop-down list is provided in the main sample module. Hard-coded options were those which could be considered more consistent, such as the purpose of sampling, specimen processing, and host information such as gender. To assist with ease of configuration, we included an Excel importer with the 6 content types as separate tabs on the sheet. In this video, I'm demonstrating the import of configurable content types from the Excel sheet. The data in the Excel document includes suggested vocabularies that exist within the template, and this includes lab hosts, host disease, anatomical material, organism, and collection devices. The users can also add to this import sheet, and we have mechanisms in place to prevent any duplication of previously imported data. To add a sample of SARS-CoV-2 and its associated metadata, the user navigates to the virus sample tab. We purposely kept this viral workflow separate in the UI to allow for easy navigation. We have base architecture fields in the default tabs and separated contextual data fields in accordance with the template. For sample lifecycle and data, we included repository accession, sample collection and processing, host information, and host exposure. The majority of fields are a simple drop-down list from configured content or hard-coded content. The mandatory fields are indicated by red squares, and the sample cannot be saved into the system without these fields being supplied. All text fields are barcode scanner compatible, and we included the sequencing tab to create a link for sample metrics and sequencing and bioinformatic metrics using the sequence library ID. Again, we had infrastructure and throughput in mind and created the virus sample Excel importer, which contains all of the fields that exist in the system. The Baobab LIMS team did, however, take this a step further and created the standalone viral analysis module. This allows for data related to basic laboratory workflows for COVID research to be entered. All 5 modules can be used together or in a variety of combinations, depending on user need and capacity. In all of the sub-modules, high throughput is taken into consideration by ensuring users can add or remove rows, depending on sample numbers for each experiment. We also captured data and timestamps, kit lot numbers, if they are being used, method linkage, and for the viral load determination, we also included some additional fields from the phage template, such as the gene name. This submodule also includes reference sample information for QC purposes. Submodules for quantification, qPCR, and sequence library prep filter samples with only the DNA or RNA sample type, so that sample drop-down lists are not a hindrance. While we still have work to do, we really hope that this simplifies an important workflow for current need. So where to next? Baobab LIMS needs user input to understand the needs on the ground. We know the system, and while we perform our internal testing very rigorously, we can miss errors. So external testing would really be ideal. We want to add metadata for bacterial samples, but we need to keep in mind that more is added increases the size and complexity of the system for end users. This may mean that we need modularity, which supplements a core. One last consideration that we need to take into account is that we should not just have data for data's sake. And this concept of data minimization is becoming more and more important when legislative principles of data privacy are applying. This means that we need to have a shift towards data by design in our digital world. So we have a number of resources available for users, and like I mentioned, we really value user input and suggestion. We also have an updated demo of the system, and you can contact me if you would like to get access. In addition, we are also scheduling a hands-on training session through the Africa CDC in April, and this will be open for anyone who is interested to attend. So I'd like to finally acknowledge the following teams and funders, and I would like to thank the organizers for providing me the opportunity to present this work. Thank you to the audience for your attention, and I'd like to open the floor to questions.